I'm going to explain thermal death point, that experiment. So here in this experiment, we're going to calculate the thermal death point of the microbial sample given, that is going to be E. coli. And at first, the theory. Well, why are we trying to find the thermal death point? Well, there is a certain temperature after which the microorganisms cannot live. Well, and why does it happen? Well, at lower temperature, um, the enzymes of the microorganisms are sort of inactive, but on increasing it, on increasing the temperature, the the activity of the enzymes keep increasing. Generally, roughly on 10 degrees Celsius increase, there is a increase, the activity of the enzymes doubles. But after a certain temperature, the temperatures get the enzymes get denatured. So after a certain temperature, the organism starts dying or stops reproducing. And thermal death point is such a minimum temperature at which the microorganism, if that microorganism is kept in that temperature, in that minimum temperature, for 10 minutes, then after that it cannot reproduce or it gets killed. And that is thermal death point. Well, that is okay. And after that, our working principle would be taking the bacterial culture in different test tubes, and then placing those test tubes for 10 minutes in a water bath, or at different temperatures starting from 50 degrees Celsius to 100 degrees Celsius, with the temperature difference of 10 degrees Celsius. Well, so there would be and after that we are going to, there would be the culture would be kept at temperatures 50, 60, 70, 80 and 90 and 100 degrees Celsius. I mean, at 100, these six temperatures for 10 minutes. And after that, we are going to place them in a sort of labeled nutrient agar plates for 24 hours at 37 degrees Celsius in an incubator. So if there is no growth, the minimum temperature at which there will be no growth would be considered as the thermal death point. Well, that is it. And then after that, there is requirements. Well, yes, but let me fix the camera later. Well, so what are the requirements of thermal death point temperature? Thermal death point experiment. First of all, we need the E. coli culture. That's it, or the bacterial culture. And then what we need? Well, we need a water bath, hot water bath, and the number of and few test tubes to give them the hot water bath. So how many test tubes do you need? Well, we need the number of temperatures we are going to give. Like here we are going to give from 50 to 100, so 50, 60, 70, 80, 90 and 100. So there would be 6 test tubes. That's it. And oh no, actually we can without using 6 test tubes, we can just use one for each. So till now we have got that we require the bacterial culture, then nutrient agar plate, and then what I was saying that so first of all, what do we need? Let's start from the beginning of requirements. So at first we need the culture of the bacterial culture, then we need a test tube, and then using the test tube, we need to give it a hot water bath. So we need the hot water bath, we need a thermometer to measure the temperatures, and then we need agar plates, petri dishes, with the nutrient agar plate, the nutrient agar, and that is the nutrient agar plate. And then and we need what is that called, loop, incubation loop or incubating loop, is to transfer the bacteria from the test tube to the what is that called, to the petri dish, nutrient agar plates. And then we need that the culture tube stand. And then what do we need? We need a incubator. um yes, we need a incubator for, allowing it to grow at 37 degrees Celsius for 24 hours. And what do we need? I guess I've said everything. I can't remember anything more. That's it, I guess. And as said, we need a culture tube stand for making this tube stand. And one more thing we need actually is the autoclave, because the nutrient agar medium actually needs to be sterilized, right? And the test tubes too. That's why we need that. And now the procedure. Yes, the procedure. And at first we need a E. coli culture prepared overnight. And then what do we need? We need to sterilize all the glasswares and the media. The nutrient agar plates. We need to sterilize them. Actually, I was trying to say that we need to sterilize the glasswares. Then we need to sterilize the media and the petri dishes in which we are going to pour the media afterwards. After the autoclave. And here actually we need a test tube for doing all the, what to say, like, for doing all the, giving all the, providing all the hot water baths. And we need actually we need 7, sorry, wait a minute. Yes, we actually need 7 nutrient agar plates, one for the control, as a control without any microorganism. And sorry, one for yes, one for control without any microorganism. And 6 for performing the experiments at different temperatures, for giving it hot water bath at different temperatures. And then here it is written that we need to give 5 ml culture is transferred from, so 5 ml of the culture, remember this, 5 ml of the culture is transferred from the test tube in the form of in front of the burner. That's it. So while giving here it is written burner, but actually we would be using the hot water bath. So 5 ml of the culture is used for the hot water bath. At first the hot water bath is given at 50 degrees Celsius, and test tubes must be labeled. The treatment is given for 10 minutes using, the temperature is monitored using a thermometer. And that's it. And we do the same thing for 60, 70, 80, 90 and 100 degrees Celsius. And after that we collect, we stick the, after each temperature we use the incubating loop to incubate the petri dish, the agar, nutrient agar plates with the bacteria, and keep them at 37 degrees Celsius in the incubator for the next 24 hours. And wait a second. And then we check. Now the checking part. See, from our previous observations we have got that, wait a second. Yes, from our previous observations we have seen that in our previous experiment we have seen that at 50, 60, 70, 80 and 90 degrees Celsius there was growth in the organism. But at 100 degrees Celsius, um, at 100 degrees Celsius, um, for 10 minutes there was no growth of the microorganism. Um, so I mean, till 90 degrees Celsius there was no growth. Sorry, there was growth, but at 100 degrees Celsius there was no growth. So the 100 degrees Celsius must be the thermal death point of this bacteria. That was the inference of the experiment. That's it. And what more? That's it. And one thing I guess I haven't written out here is the control. Control, I think, guess I should have written out here.